Yeah, there will be a Q&A in the end. So if you want to ask questions, please put it in the chat box, and I will then collect all of the questions and will hand them over to Aishwarya. So yeah, this brings me to the favorite part of my evening now, which is introducing Aishwarya. So I'm very, very happy to introduce her. She's a postdoc fellow and a friend of mine, so Dr. Aishwarya Jacob. And many of you will know her because she has been a postdoc convener for a while. She's introducing the talks here, doing a lot of stuff around the college, and she's there quite a lot as well. And today she will be talking about her journey navigating academia from India over the US and landing in the UK, and her research, and possibly dinosaurs. I have heard. So there's one already behind me, listening very excitedly. And I think we're all looking forward to Dr. Aishwarya's talk. And yeah, over to you, Aishwarya.
Thank you, Eva, for that lovely introduction. Let me start off by saying that I do not have data on my progress on cloning dinosaurs, but I'm hoping that will come up pretty soon. So maybe the next Lucy in lockdown. I'm making my way there, so trust me. Thank you again for the opportunity to present at Lucy in Lockdown. As Eva mentioned, I've hosted a few of these series. I've had the honor of hosting very wonderful speakers before me, and I'm a little bit nervous about what I'm going to be telling you about, but I hope you will enjoy my tiny little tale of how I made it here and how I'm with Lucy. And I'm going to share my screen now. Okay, I hope you can all see my screen and my little cursor. So essentially what I'm going to do today is give you an extremely brief but excruciatingly detailed and exhaustive, in-depth overview of how I got here and what actually I'm doing here. But I don't think I can tell you why I'm here again, because I'm a terribly confused postdoc, and you will see why in a little bit. As Eva said, I'm Aishwarya Jacob, and again, thank you for this opportunity for presenting at Lucy, not anymore, but in Lockdown series today. I started my scientific journey in India. I was born and raised by this pair who are attending the talk right now. Hi Ma, hi Pa. So as I said, I was born and raised there. I did my schooling in Coimbatore, which is the city where I was raised. I also finished my bachelor's degree at the Anna University in Coimbatore in the PSG College of Technology, my specialization being industrial biotechnology. And at the end of my 4-year degree, just like most other undergraduates, I had very specific opinions, and one of them being exactly how I was going to lay out my career, what I was going to work on. I had very, very specific opinions. I was going to work exclusively on cancer biology. It didn't matter what exactly was within the realm of cancer biology, but it was going to be involve cancer biology, preferably molecular biology. I was pretty open-minded about that. Gene transcription, epigenetic control, protein biology, you name it. I'm very easy, but never this particular arena called RNA processing. For those of you who are not familiar with what it is, I will enlighten you in a few minutes. However, suffice it to say that at the end of my undergraduate degree, and also from my experiences in high school, RNA processing was something that simply did not work for me, either in theory as well as in the laboratory. So no, yuck, I will not touch it. At any rate, in 2008, fresh out of college, I did something entirely unexpected. So unexpected that even now I'm completely like, wait, how did I do that? First thing was that I actually left home. I'm a bit of a homebody, and the fact is I decided to spread my mission to cure cancer and took it to what became Obama land, and I went to the Ohio State University. And during my graduate career, under the supervision of Dr. Don Chandler, I continued to work on cancer biology. And about 6 years later, in 2014, they told me, you can actually call yourself Dr. Aishwarya Jacob. And that made me think, oh, therefore I must be fully qualified to eradicate cancer. So if you ask me what I actually did to earn that PhD, I will elaborate a little bit more, but to be honest, I'm not entirely sure. But suffice it to say that it actually ended up being phase 1 of eating my own words, because you see, I specialized in RNA processing, the one thing I said I will never do. But the consolation is that it was still in cancer biology. I worked on a form of childhood cancer called rhabdomyosarcoma, and also in lymphoma, which was a disease that took my grandmother many years ago, and which was actually one of my primary reasons for getting into cancer biology in the first place. So yes, I'm still sticking to my guns. I'm in cancer biology. So with my new PhD, I flew off to UC Santa Cruz, University of California, all the way across to the west coast of the USA, and started to engage with the banana slugs over there to cure cancer by the sheer power of basic molecular biology. And I worked with Dr. Jeremy Sanford from about March 2015 to middle of 2016. Here I worked more on breast cancer models, but I really needed a change. And trust me, it was not that I was trying to run away from anything. I just really needed a change. So 2016 was a year of many changes for all of us. That was when Obama land became that land. And then I said, fine, I'm going to leave. And I walked straight into Brexit. But apart from that, another major change in my life was that I had to eat my own words once again, because you see, in 2014, I am solely interested in cancer research. And now I work with cardiovascular biology. So you see, I had to leave the field of cancer biology. I say I had to leave. Honestly, it was a choice. And you'll see why, because I will highlight to you why I enjoy my science so much, that I do not regret having turned away from cancer research for the moment. Not only did I acquire a new field of research, I acquired not one, but 2 new bosses. The one on the left is Professor Chris Smith of the Department of Biochemistry. He is a hardcore basic biochemist, the type that can draw amino acid structures on the fly. Remember, their charges, how they migrate in a gel, all without the use of Wikipedia. And on the other side, you have his polar opposite, Dr. Sanjay Sinha, who is a hardcore cardiac surgeon, and that man in the room who will ask you, well, why does this matter again? But I will give a shout out to my boss. He definitely is a visionary in the sense that he's very keen on linking bench science to bedside science. And bridging these 2 gentlemen is me, one confused postdoc. So you might ask me, well, what exactly is the science that you actually do? And in order to explain that, I shall have to take you back to the central dogma of life, or gene expression. Now, gene expression, as we all know, begins with the hit single by the K-pop band BTS called DNA, and then works its way into all these healthy foods that we're supposed to be eating way more of in order to lose weight. Right? That is really all that matters in the central dogma of life. However, what a lot of people don't appreciate is that there is an intermediate dark horse stage in the form of this molecule called RNA, or ribonucleic acid. Ribonucleic acid apparently is also available as supplements, as shown here. Do not take them. I think they're a hack. I just had to warn you about that. But let's break this down and make this a little bit simpler, shall we say, and take a molecular perspective of the whole thing. So DNA, which is, just to simplify matters, is compartmentalized into what are called genes, is where it all starts. Now, the DNA is basically a code that eventually gets translated into a protein. And different genes will make different proteins. And what we call as protein in our foods is simply a mishmash of the different types of proteins. So originally it was believed that one gene makes one protein, and I will tell you how that is not the case in a couple of seconds. However, what happens is that DNA undergoes transcription, where this code is transcribed by a set of protein machines into what are called the messages, or the messenger RNA molecules. And this will also define how much of the protein you make, because you can make multiple copies of this straight from the gene. This mRNA is then taken out from the nucleus where this entire process occurs, and it's translated. The code is further translated at one more stage by these huge machines, molecular machines within the cell called as the ribosomes. And what they do is string together a bunch of other molecules called the amino acids to make the polypeptides, which will eventually assemble into proteins. Simple, right? And really, proteins are the end product, or what we think are the end product of what we see in terms of gene expression, and these are the things that actually affect the functioning of the cell. While that is true, that is not 100% true. You see, what you're actually missing is a crucial middle piece. I think just for the fun of it, evolution decided that it's going to interrupt a gene by putting in bits of DNA that actually just don't seem to mean anything. They call it the junk DNA. So you've got these exons shown here in the blue boxes. Those are those are actually the ones that constitute the final message, which will eventually become the protein. These bits of DNA in between, the junk DNA, actually have to be removed in a process called a splicing in order to make the final messenger RNA. And if you don't remove it, you end up with a lot of problems that we will not talk about just yet. So simple, right? Here you've got your gene. You've made what is called as a pre-mRNA. Easy peasy, I'm going to cut these little bits out. I'm going to make a message, and then we get the protein. And this is where I'm going to make life a little bit more complicated. There is a process called as alternative splicing. And what this really means, without going into too much detail, is that there are several molecular machines within the cell that can help you pick and choose which bits of the DNA are encoded into the message and which bits will finally make up the protein. So essentially, what you get from a single gene is not one protein, as was previously surmised. You're going to get an array of different types of proteins. Pretty economical, right? You've got one long message, but you can make multiple different end products out of it. So it's a really neat way of packaging a lot of information into a relatively small genome. So these proteins are now called as isoforms, and what you're getting is multiple isoforms from a single gene, and this is what really affects the way your cell functions. So what does alternative splicing actually mean for us? So what that means is that it can actually affect the way a cell makes decisions in various processes. For example, a switch in the type of splice variant, as we like to call them, the different kinds of messages you make from the gene, will determine whether a cell is to live or die, for example, in apoptosis. Aside from that, it is a heavy contributor to tissue diversity or cell diversity. Essentially, alternative splicing will tell a brain to be a brain, or a neuron to be neuron, to be muscle cells to act as muscle cells, and to behave like muscle cells. So gene transcription is an important component of all of this. I will not downplay that, but just as important is the process of splicing. Finally, and quite importantly as well, splicing plays a major role in disease. Sometimes it's causative, and sometimes it's actually resulting from a series of other processes, and that can contribute to disease progression. Common examples are various types of cancer, which have aberrant splicing in them, and also different types of muscular dystrophies. Just to sort of put this in perspective, your question to me might be, well, can we actually modify the splicing process now that you tell us it might be important disease? And the truth is, yes, there are several techniques, including the famous CRISPR that we all know is commonly used to make genetically engineered human beings from babies. You can use CRISPR to alter splicing as well. However, what's actually made out successfully as therapy is this other technology called as antisense oligonucleotides, or AONs. These are short bits of molecules similar to the RNA. What they can actually do is go into the cell and combine or bind different parts of the pre-mRNA and can help determine which splice variant or which messenger RNA is finally being made, which in turn, as I said, will determine the type of protein you make from that gene. Just to put that in perspective, I'd like to take a couple of seconds to give you an overview of the tale of spinal muscular atrophy. SMA is a neurodegenerative disease that specifically affects the motor neurons. So what that means is these are the neurons that are helping you that innervate the muscles, the skeletal muscles of your body, and help you to control them. So in patients, in patients of SMA, they do not have a gene called as SMN1. It's completely non-functional. So what happens is they lose functioning of their motor neurons, which eventually leads to muscle wasting. And these people, in varying degrees of severity, people end up losing usage of their muscles, and sometimes it leads to, actually quite often leads to fatalities. So seen here is a child that was diagnosed very early on in life and was almost completely wheelchair bound. So the reason spinal muscular atrophy really happens or really occurs is, as I mentioned, because they don't have the gene SMN. Most of us have another gene called SMN2, which in theory can compensate for SMN1. However, unlike SMN1, it cannot splice in this particular exon 7. So the normal message that you would make out of SMN2 gene is this particular message, which makes a shorter protein, SMN delta 7. So if you had the normal SMN1 gene, then you wouldn't feel anything at all. You wouldn't miss this gene at all. However, for these people who don't have SMN1, they rely on SMN2 to make some amount of SMN protein, which is very critical. So decades of research by Dr. Adrian Craner, who's currently at Cold Spring Harbor, showed that you could actually alter splicing in the SMN2 gene simply by using an oligonucleotide that will force the inclusion of SMN exon 7. So what that means is you're now making fully functional protein even from a gene that is normally dysfunctional. And this has resulted in the first FDA approved, I think it was back in 2016, the first FDA approved splicing correction therapy called nusinersen or Spinraza. And here you can see this child has been given this injection and he is now able to stand up almost unassisted. I am told that this gentleman makes people cry at the end of his presentation in great emotion. I promised myself that I will make people cry someday at the end of my talks. I hope it will be all of you today. So in order to make these drugs, you first have to understand how splicing actually occurs and how it can be controlled within a cell. And you can think of splicing, the process itself, the process of removing this junk bits of RNA, as an intricate waltz that happens between the pre-mRNA that is being processed and accessory proteins or controller proteins. The basic executor machinery, which is required for every single splicing event, is called the I will not go into the details of that. However, suffice it to say, this is a multi RNA protein gigantic complex called the spliceosome. Now, the process of alternative splicing introduces an extra layer of complexity to the whole process. And what you actually need in order to decide the fate of an exon, be it whether or not to include it in the final message, is the action of a certain class of proteins called the regulatory RNA binding proteins, or we will call them RBPs. When I say they bind, what they can do is they can actually recognize signature binding elements or codes on the pre-mRNA, and they will simply form chemical connections with that, and they will sit on the RNA and perform a series of chemical reactions that will eventually decide the fate of the exon. It's been 4 decades at least since we have learned about alternative splicing, but I can safely assure you that it's still very poorly understood, and that's why I still have a job. So when I joined graduate school, I became a splicer. And what I studied was the alternative splicing of a famous oncogene called MDM2. You can actually think of MDM2 as a gremlin. For those of you who watched the movie, you know, they're cute. You have one gremlin, they're cute. You know, that's normal splicing for you, keeps things under control, all lovely and wonderful. Then you feed them after midnight, or I suspect you also pour water on them. You get too many of them, and that's when it becomes beastly. And that's exactly what MDM2 is like. That's why it's an oncogene, because when there's more copies of it, it contributes to cancer. What I worked with was what I called the anti-hero of it all. Alternative splicing of this very complex gene can lead to the production of a much smaller protein called ALT1, which at face value is actually beneficial to the cell and protects you from the gremlins. However, under unknown circumstances that I discussed quite extensively in my thesis and would not care to revisit at the moment, it can be pushed into villainy, and what you end up getting is a tumor. So my dissertation work addressed how is ALT1 being made at this level, and with the potential for developing strategies to actually block the production of the message of ALT1 itself. And the second part of my dissertation was trying to understand how this protein that you get from this aberrantly spliced message, how does this ALT1 protein actually cause cancer, with the implication being that if we could figure that out, we could actually block the activity of ALT1. And if you're very interested in it, I will give you the references to my papers, but shall we say it's not entirely clear still. So with that experience, I went to my first postdoctoral project at UC Santa Cruz. I shifted gears just a little bit and went into using a breast cancer model. And what I wanted to understand at this stage was a little bit more basic. What I wanted to see was if changing the splice variant or the messenger RNA that you make, will that also affect not only the type of protein you make from that gene, but also the amount of protein you make from that gene, with the idea being that in cancer cells, when you have a splice variant that is not particularly good for a normal cell, but actually absolutely amazing for a cancer cell, you would simply end up making not only more copies of that splice variant, but also more copies of the protein itself at the level of translation. And my goal was to figure out this mechanism so that I could actually take those antisense oligos that I told you about just now and block this process so that I could try and revert the cancer cell status back to a normal cell status as best as we could. So that was my first postdoctoral experience. But then, as we know, I shifted fields and decided to go into cardiovascular biology, but I'm still a faithful splicer. So my current research is actually a bit like figuring out how Dr. Jekyll became Mr. Hyde and how Mr. Hyde goes back to becoming Dr. Jekyll. So that's literally what it is. So Dr. Jekyll here is a very niche cell type called the vascular smooth muscle cell. I will call it as a green cucumber for purposes of scientific clarity. This green cucumber that you see here is actually a cell that lines the walls of your blood vessel, and shown here is your blood vessel wall, typically. So these cells, these sort of oblong, elongated cells, are very important because they make up the structural integrity of your blood vessel wall, and especially in the major arteries like your aorta, for example, which is practically a hose pipe for blood. But importantly, what it also does is that it expands and contracts and makes sure that your blood is flowing properly and your blood pressure is being maintained. So, for example, hypertension, which is something my dad has, can actually often be caused by hardening of the arteries, artery walls, or also, shall we say, a misfunctioning of these particular cell types. So that is an example of a pathology in which vascular smooth muscles are involved. So a typical characteristic of these green cucumbers is that they're very well defined in their structure, and they are very contractile, which means they can contract and relax. And what they also have are high levels of what we call smooth muscle cell markers, which are just proteins that will tell you that a cell type is a specific cell type. So under certain conditions, which include having way too many late night McDonald's or smoking, genetic risk factors as well, I will acknowledge that. What happens is you end up causing a phenomenon called as phenotype switching because of insult or injury to the wall of your blood vessel. So what I've shown here is an example of an atherosclerotic blood vessel. So you have this plaque here, which is an accumulation of lipid and various other materials that will occlude or block your artery and prevent blood from flowing through. And that can cause many complications. Depending on which artery you're talking about, you can either get a stroke or a heart attack, right? So smooth muscle cells, the vascular smooth muscle cells that I'm working with, actually play an important role in this. In the first line, in the first reaction to the insults, such as accumulation of fatty acids or lipids, what they do is they morph, or what we call phenotype switching in the lab. They go from these beautiful green cucumbers to blue Doritos. And these blue Dorito chips, as we see them, are not really good for you, although they're an important defense mechanism. Over time, they morph themselves into other cell types as well, and eventually contribute to forming the plaque. So what started off as a good thing has morphed into something that is not ideal. So these blue Doritos have several characteristics unto themselves. One, they're not contractile. They secrete certain what we call proteinases, which are like scissors, if you will, that can cut up all of these scaffolding proteins that are holding all these guys together, and can actually break through and get into the within the lumen of the blood vessel. Apart from that, they're very proliferative, which means they can divide quite endlessly, and they're also very migratory, which means they can move around. While these guys are fairly static, the green cucumbers, these guys like to move around, the blue Doritos. So when this happens, who are you going to call, right? You have to call upon people at the Smith lab. So what we do in the Smith lab is quite unique, actually. We do not care about the gene transcription and the gene expression being controlled at that level that all the other, run-of-the-mill kind of scientists do. No, we're exclusive. We study alternative splicing. So what is known is that genes that are exclusively expressed in vascular smooth muscle cells, which tell a cell that, yes, you are a vascular smooth muscle cell. These genes undergo alternative splicing. And what you can get are these green isoforms in the green cucumbers called the SM isoforms. And that relies on selecting which specific bits of the VSMC genes to be included in the message and eventually in the final protein. when the cell's phenotypes switch into the blue doritos, you end up making a different choice of exon. And what that makes is a completely different protein. So these two proteins, like I've said before, come from the same gene, but they have vastly different functions. For example, a protein called tropomyosin, the smooth muscle isoform, is the kind that will help the cell to contract well, whereas the non-muscle isoform of the same tropomyosin protein will not allow the cell to contract as well as it should. So my job is to actually figure out how smooth muscle splicing is being controlled. So we know it happens. And the question now is, how is it actually being controlled or regulated? And also, what is the extent to which smooth muscle splicing actually determines the behavior of the green cucumbers and the blue Doritos? So when I started out in the lab back in 2016, we had found a legendary regulator called, with an absolutely imaginative name. It's called the RNA binding protein multiple splicing, or RBPMS. Now it's called an RNA binding protein because it binds RNA. It's called multiple splicing because this protein is itself subject to the process of alternative splicing, making different types of RBPMS proteins that have very different activities. I will not get into that because that is a whole other 500 hours worth of lab meeting material from the Smith lab, so we will not go into that at all. What we do know is that RBPMS is highly expressed, which means there's multiple copies of the RBPMS protein present in the green cucumbers or the differentiated cells. However, the proliferative blue Dorito smooth muscle cells lose the expression of RBPMS. So the hypothesis that we started out, and which we have proved more or less, is that it is a tissue-specific master splicing regulator. The master is just, we've actually published this word. It's pretty amazing, really. It's a master splicing regulator that is very critical for the identity of these smooth muscle cells and also their functionality. So previously, the findings from our lab have been in the rat model. Who cares about rats? So let's get into the human system. Because adult humans are somehow very reluctant to part with their blood vessels. I don't know why. It's very strange, really. I'm being forced to work with what are called the embryonic stem cells. So this is a typical colony of embryonic stem cells, and as most of you have probably heard, stem cells have the ability to pretty much differentiate or make any type of cell within the body. So provided you give them the right chemical cues. So Dr. Sanjay Sinha's lab, after years of blood, sweat, and tears, came up with a very streamlined protocol that over the course of 17 days can turn these embryonic stem cells into smooth muscle cells. And so I've shown you just a quick picture of one of my differentiations, and these are the cells that I get at the end of about 17 days worth of work, during which I have to go in every single day and baby them. These cells are glowing green because they've been marked with a certain protein called as the green fluorescent protein, which will come into play in a couple of slides. I'll talk about it more then. Suffice it to say that the smooth muscle cells I make out of the embryonic stem cells are very immature, and that's why I've colored them blue and not green, because green in the course of the schematic of this presentation would be more or less a differentiated cell. So these are very immature, very proliferative, and actually closer to the blue Doritos that I have been talking about all along. So what I wanted to understand was, can we manipulate splicing in these cells? And that to splicing in the context of RBPMS, and see if I can understand how splicing can impact the transition from a blue Dorito to a green cucumber. So the model system that I've used is an inducible smooth muscle cell model. This diagram is very intimidating, even for my bosses, so do not worry if you don't understand it. Just suffice it to say that I can make smooth muscle cells, and when I add a certain drug called doxycycline, when I make a certain, when I add a certain drug called doxycycline, either my cells will glow green because of a marker protein called as GFP. Think of GFP as a light bulb. I'm borrowing this analogy from one of my colleagues, actually. So think of it as a little light bulb. So as soon as I've added the drug or the switch, the light bulb turns on, and I know the gene has been activated. So this is my control cell in which only the GFP is being expressed. So these are my test cells where I know that if I'm expressing GFP alongside that, I'm expressing RBPMS. The reason this has to be done is because the cells that I make from my embryonic stem cells do not have RBPMS, and this is a key indicator of how immature they are, because remember, I told you that the mature cells from a tissue will have RBPMS. So this becomes an ideal platform for me to see how RBPMS can manipulate splicing and therefore the functioning of these muscle cells. So don't worry about this. But this is, these data that I've shown here are simply to show you that I can actually separate the cells that are green and those that are not green in color within the same plate, and that will tell me whether or not I have RBPMS in them. So what I have done is I've started with the blue Doritos. I've put RBPMS in, and my question is, can I command these cells to become green cucumbers from the blue Doritos? That is, go from an immature state to a mature differentiated state. Again, this is a very complicated piece of bar graph. But let me assure you that the taller these bars are, and for those of you who are more interested in the technical aspects of it, I'm glad to elaborate later. Suffice it to say that the taller the green bars are, the more it's an indication of how RBPMS has made my cells more mature. Just to sort of bring that into perspective, what you're looking at is an experiment called RT-PCR. Now, this is a direct indication of the types of mRNA and therefore the type of protein that will be present in a cell. What I can show you is that the control cells where I don't have RBPMS, you only have a certain type of protein being made that is not functional in, that is not present in the mature smooth muscle cell. However, when I throw RBPMS in, I now get this particular isoform. It's called as metavinculin here. Don't worry about the name, but suffice it to say that metavinculin is a protein that is indicative of very advanced mature cells. Not only that, this is a protein, or the protein that is made from this RNA is very important in helping a cell to contract and relax. So what I've actually ended up doing is making cells with RBPMS that is somehow pushing them to have splice patterns similar to the mature tissue. So this is to say, this image is not for you to sit and stare at and analyze. That's a job for Chris, my boss. He's doing that as we speak, I believe. What is important is that I want you to understand that by profiling the cells through a technique called RNA sequencing, RBPMS actually affects the splicing of over 600 genes and therefore produces several different isoforms from over 600 genes. And these proteins that are so different can actually affect the way the cell functions. And what we can actually do is we can look at these different genes. All of these are a subset of genes that are being affected by RBPMS, and you can see how they're all connected. So it's not just an isolated gene, but these are all connected into networks as well. So when you affect splicing, you affect specific networks so that a bunch of genes as a group can affect the way a cell behaves. So some of the classes of proteins that we pick up are cytoskeleton, which are basically the proteins that hold the cell together structurally. Focal adhesion proteins are those that help the cell to stick on to their surface, and which are very important for them to stay in place, as well as to form their shape, to move, etc. And finally, and most importantly, a class of proteins called the contractile proteins that are important for performing the function of contractility, which I told you is a critical function in your blood vessels. So yes, so RBPMS can make blue Doritos into green Doritos. But the question is, unless I've altered the splicing, but unless I can actually detect different behavior in my cells, I cannot tell you if they've actually become mature green cucumbers or not. So that is something I'm currently trying to do. And what I've shown so far is that indeed RBPMS, the presence of RBPMS, makes my cells divide less or proliferate less, which is an important characteristic of mature tissue smooth muscle cells. It also makes them move around less, which is another important characteristic. But I guess the sort of holy grail of all features is how well can my cells contract. And that is something I'm working on and have been actually for the past 3 years to see if RBPMS can actually modulate that or not. So just to sort of bring you back, what we have found is that splicing can indeed impact the way smooth muscle cells behave, at least in vitro. And RBPMS seems to be quite a prime player in the sort of transition between your good Dr. Jekyll and your crazy Mr. Hyde. And what we want to know is, since all of these experiments have been done on plates and in vitro, as we call it, we want to see if we can actually see this sort of behavior in vivo. And again, humans are somehow very reluctant to let people experiment on them. I don't know why. What we're forced to do now is actually work with mice. And what we're trying to do is build models, either of atherosclerosis, which would, as I've shown you before, the cardiovascular disease, or an easier model to work with is an injury model. What we do is we can take mice and we can injure their carotid artery, which is the artery that sort of goes all the way up to your brain. Just to put it in very simple terms, what we do is we injure that artery, and we look to assess the properties of the artery post injury. What I can tell you is that when the wall of the blood vessel is injured, you lose the expression of RBPMS, and you also observe changes in splicing. So these are events that are going hand in hand, which gives us a lot of confidence to actually move forward in asking the questions about how RBPMS and the splicing networks that it directs are important in sort of cardiovascular pathologies. If you think about it, you have the cardiovascular disease, but also a process called restenosis, which is what this injury model is actually trying to mimic. The injury to the vessel wall is quite critical, and if you're actually able to modulate splicing at that level, the sort of potential is quite endless. So in the future, and in the very near future as well, what we want to do is manipulate RBPMS in adult mouse arteries. And this was actually a collaboration that was supposed to happen at an institute in Spain. And obviously the pandemic, which I shall not mention again, has disrupted these plans. However, we hope to resume those experiments very soon. Aside from that, the other goal is to genetically manipulate the splicing in adult arteries again, so as to understand how this entire process of pre-mRNA splicing and its regulation can affect the structure and functioning of blood vessels, especially the arteries. So I have to thank the British Heart Foundation mainly for funding my work, and also currently the Wellcome Trust grant, which funds me and lays food on my plates. But let's just take a step back and ask ourselves why the British Heart Foundation should actually care about my very basic research. Unless you understand how your normal cell functions, your normal tissue functions, we are not going to get a sense of how they're disrupted in disease. And as you can see, this is just a small sampling of the sort of cardiovascular diseases one can acquire in life. And believe me, every time I leave lab meeting in Sanjay Sinha's lab, I feel like I have a new disease. So what you can appreciate is that splicing, there are several lines of evidence to suggest that pre-mRNA splicing is involved in many of these pathologies. And if we get an in-depth insight into how this process is controlled in normal heart and in the normal blood vessels, we can potentially extend that knowledge into developing novel drugs that will eventually target, that can target pre-mRNA splicing. So with that, I'd like to thank the Smith Lab. And this is my boss, Chris, and these are some of my lab mates who've been very instrumental in getting me to where I am today. And the Sinha Lab, this was us just before the lockdown, going to the Crystal Maze. See, we can actually stand next to each other, which is crazy, right? I would never do this again. Anyway, with that, thank you for your attention, and I'll be happy to take questions.
Thank you so much, Aishwarya. That was brilliant. Are there any questions? No. I have loads of questions. So if nobody, like, please put your questions in the chat box. Even like if you want some point clarified, or are just curious about the future, please put it in, and that's a valid question as well.
Yeah, don't worry, it doesn't have to be a scientific question. You can even ask, exactly, why is your hair looking like this today? It can be anything.
Or what is this and this good for, or whatsoever. And like, I'm a plant person, so I'm gonna probably ask really stupid questions as well.
Okay, I'll take your questions as stupid.
Okay, cool. Like, whilst everybody else is still thinking about their questions, maybe I can ask some questions. So like, so you have the blue Doritos and the green cucumbers, which I love. That's a good question. So what are the blue Doritos for? Is it just for repair? So like, it goes back to embryonic for if it's hurt, and then it needs the blue ones, yes, to proliferate. Okay, cool. And yes, if you so that's really interesting. It's really similar to plants. And then if if you take out the blue Dorito axon, what happens?
We don't know that yet. That's something we really want to find out. So okay. What that really needs is an extra pair of hands, 2 other postdocs, and a few research assistants. But yes, you always need more. So yes, that is definitely a question that we're trying to ask. So what if we force the expression of the non-muscle isoform, or we force the expression of the smooth muscle isoform in places where they're not supposed to be? How are you going to fix that? So it's a lot like taking the spare parts of a Maserati and trying to put it into a Ford and trying to see if it will run or not. So yeah.
And about the cell specificity, is it just via the RBPMS, or are there other factors involving that?
So there's several other factors involved in it as well, and that's something we have some other lines of evidence for as well. So it's not just splicing factors, it's also transcription factors. I've really downplayed that today. But yes, there are several other factors involved, but we think that RBPMS was one of the important players. Think of it as a drug ring. You've got the drug lords at the top. There's never just one drug lord, right? You've got quite a few up there. So yeah, so it's not the thug at the bottom of your drug chain. It's one of the drug lords. At least that's what we're trying to tell the publishers.
Okay. So Henriette has a question. And Anke, the Ankylosaurus as well. What happens with the dinosaurs? We were promised dinosaurs.
I think I need to change bosses in order to make dinosaurs. And at the moment, I'm happy with both of mine. But maybe for my next assignment, I will look into that. But that being said, if I get dino DNA, I can make dinosaur muscle cells. And I can study splicing in them. I can tell you that much, and I'm very confident of it. So,
of course, you can avoid them having heart disease.
I can avoid dinosaurs having heart disease. Yeah, yeah. So one cell type at a time, but we'll get there.
Are there any more questions? If not, I'm gonna hand over to Henriette, who wanted to have a little comment as well.
Yes. Yeah, if there is indeed no more questions, then I'll just do that right now. Because Aishwarya, we're just using this possibility, this opportunity rather, so that we can say a little thank you to you. Not all of you who are listening will know this, but the title of this series is actually Aishwarya's, Lucy in Lockdown. She came up with this. And this is just one of the examples of all the various things that she's been doing with the college and for the postdocs and for the whole college community in the last few years, in her role as postdoc representative. I've worked with Aishwarya with lots and lots of pleasure. She is really a resourceful person. She is an absolutely enthusiastic and original thinking person. She's a people's person. She's a great organizer. And as you will have seen tonight, she is a great communicator who uses lots of humor. And the humor was definitely there tonight as well. So I will never, ever open my fridge and look at one of those really gone bad green cucumbers. That's about the only vegetable that ever goes bad in our fridge, but that one. And not think that maybe it's just a vascular smooth muscle cell that has gone blue Dorito on me. So that was, you know, it's... So on behalf of the college, I'm just taking these 2 minutes to say a huge thank you to you, Aishwarya, for all the work that you've done for the postdoc community, but also for the community at large, because I think everyone in the college is now aware of the fact that there are postdocs. They are visible. And you've been very open to working together with the fellows and also with the grad students, and that has just provided that little glue that brings all the groups together, and it's been absolutely fantastic. It will be absolutely fantastic to work with the people who are following, including Eva and Christina, but once again, a very big thank you. And there will be something in your pigeonhole at some point in the college. Don't go and run there tonight because I haven't exactly organized it, but there will be something there, and I'll send you a quick email to say it's there as a thank you, just from me personally. Okay.
Thank you. It's such a pleasure working with you, Henriette, and of course, Lucy is such a wonderful community, and I'm incredibly grateful for being a part of Lucy, and of course, I'm continuing to be with them. And very excited to see where all of this goes. Thank you again. I really appreciate the opportunity for letting me work with all of you guys these past couple of years. Appreciate it. Thank you. Very kind
Thanks
words.
as all ours.
Thank you.
Eva, back to you.
I think we are basically done now. Thank you everybody for joining. Thanks so much for your talk, Aishwarya. I still have so many questions, but I will ask you directly.
We'll talk later.
Definitely. Okay, cool. Thanks everybody for being part of this. If you have more questions that you're coming up with later, just send a message and we will forward it to Aishwarya if you don't catch her directly. And I'm sure she's happy to talk about this. Yeah, thank you everybody. Bye-bye.